All right, let's jump right in. Today, we're going to talk about a really fascinating but also pretty complex disease called amyloidosis. It's one of those conditions where the body's own building blocks, well, they start to go rogue, causing this huge cascade of problems that all starts at the tiniest molecular level. So, picture this. Your body is like this incredibly intricate machine, right? And it's built from these microscopic parts called proteins. Each one is folded into a perfect, super specific shape to do its job. But what happens when those little building blocks get bent out of shape? And that, right there, is the heart of amyloidosis. For reasons we're still trying to figure out, certain proteins they lose their proper shape. They get all sticky and start clumping together, forming these things called amyloid deposits. Imagine gunk just gumming up the works in a super important machine. Except that machine is your heart or your kidneys or your nerves, and when they get clogged up, it messes with how they work and, yeah, that leads to some really serious health problems. Okay, so here's the first big twist. Amyloidosis isn't just one thing. It's more like an umbrella term for a whole family of related diseases. To really get a handle on it, we gotta meet the main players. Because figuring out which specific protein is causing all the trouble, well, that's the #1 clue for doctors. All right, let's look at the two most common types you'll hear about. AL and ATTR. The huge difference between them is where the problem starts. With AL amyloidosis, the issue is with something called light chains. These are pieces of antibodies that come from plasma cells in your bone marrow. But with ATTR, the culprit is a completely different protein called transthyretin, or just TTR for short. You can see how knowing which one is misfolding is like the most important first step. And believe it or not, the family's even bigger than that. You've got other types that can be kicked off by things like chronic inflammation or even some long-term medical treatments. But no matter what, the core idea is always the same. Some specific protein is misfolding. And finding that protein is always step one. But what if the clues start pointing to more than one troublemaker at the same time? And this is where things get really interesting. And honestly, really challenging for doctors. It's a true diagnostic puzzle. What happens when different types of amyloidosis decide to show up in the exact same patient? So that brings us to the big question, right? Is it even possible to have both AL and ATTR amyloidosis at the same time? And this isn't just some academic question. The answer can literally mean the difference between life and death when it comes to treatment. And the answer is yes, it is possible. Now, it's considered rare, but it's not so rare that we can ignore it. Some estimates say that in maybe 5 to 10% of amyloidosis cases affecting the heart, you could find both types of these protein deposits. You can imagine how this makes getting the diagnosis right not just complex, but absolutely critical. So here's the problem. The symptoms, things like being tired all the time or even heart failure, can look exactly the same whether it's AL or ATTR. They're identical. That's why doctors can't just go by how a patient feels. They have to bring in the big guns, super precise tests like mass spectrometry on a tissue sample. Think of it like a molecular scale that can identify every single protein involved. It is pretty much the only way to solve this puzzle for sure. Okay, so let's zoom in a little bit. We're going to take a deeper dive into just ATTR amyloidosis for a second. Because it turns out, even within this one type, the problem can actually start in two totally different ways. So you can see the two paths right here. On one side, you've got what's called wild-type ATTR. In this case, the TTR protein itself is totally normal, but it just starts to misfold as a person gets older, maybe due to the stresses of aging. Then, on the other side, there's hereditary ATTR. And here, a genetic mutation means the liver is actually making a faulty, unstable version of the protein right from birth. And as you're about to see, knowing which one it is, well, it changes absolutely everything. So you might be wondering, why does all this detective work matter so much? What's the big deal? Well, it's because figuring out the exact type and subtype of amyloidosis is the only way to get to the right treatment. And I mean, the strategies are worlds apart. For AL amyloidosis, you have to go after the source, the plasma cells making those bad light chains. That often means treatments like chemotherapy. For both types of ATTR, there are these cool stabilizer drugs that basically act like a brace to keep the protein from falling apart and misfolding. But then, and this is key, for hereditary ATTR specifically, there are incredible gene silencing drugs that can actually tell the liver, hey, stop making so much of that faulty protein. And this right here is probably the most powerful example of why getting the diagnosis right is so critical. For someone with hereditary ATTR, a liver transplant can actually be a solution. And the logic is simple. The liver is the factory pumping out that faulty protein. So if you replace the factory, you stop the problem at its source. It's brilliant. But, and this is a huge but, a liver transplant would be completely useless for someone with wild-type ATTR. Why? Because their liver is making a perfectly normal protein. The factory isn't the problem. A transplant would do absolutely nothing. So, if there's one thing to take away from all this, it's this. In the super complex world of amyloidosis, everything, and I mean everything, comes down to identifying exactly which protein has gone wrong. That single piece of information is the key. It unlocks the diagnosis, and it points the way to the right treatment. You know, a disease like this is just a powerful reminder that sometimes the absolute biggest problems we face start with the tiniest little errors on a molecular level. It really makes you wonder, doesn't it? What other huge biological mysteries are out there, just hiding in plain sight inside our own bodies, waiting for us to figure them out?